I'd like to welcome everybody to the first lecture in the IGB Distinguished Speaker Series for this academic year. The lecture today is co-sponsored by the new Center for Biomembrane Systems here at UCI, directed by Hughes. And it's a great pleasure to welcome Andy McCammon today. He got his BA from Pomona College, MS and PhD in chemical physics from Harvard University. In 1976 to 78, he was working with Martin Karplus at Harvard and developing some of the first computer approaches to simulating proteins and protein dynamics. He then went to the University of Houston as an assistant professor of chemistry in 1978. And he became the Anderson Chair Professor of Chemistry in 1981. In 1995, he moved to UCSD, where he's currently the Joseph Meyer Chair Professor of Theoretical Chemistry and Distinguished Professor of Pharmacology. He's also an HHMI investigator. Professor McCammon has invented theoretical methods for accurately predicting and interpreting molecular recognition, rates of reactions and other properties of chemical systems, for simulating proteins, and for discovering, screening new drugs in silico. He has authored or co-authored over 600 publications in theoretical chemistry and biochemistry. And he has worked also with the industry in the 80s. He worked with Agron Pharmaceuticals, which is now Pfizer in La Jolla, and contributed to the development of some widely used drugs, such as Viracept, which is an HIV-1 protease inhibitor. More recently, he has worked on a drug which was approved in 2007, which is an antiviral called Isentress, which is distributed by Merck. He has received many awards, too many of them for me to list here, but I'll mention the George Herbert Hitchings Award for Innovative Methods in Drug Design from the Burroughs Wellcome Fund in 1987. More recently, in 2008, he was the recipient of the American Chemical Society National Award for Computers in Chemical and Pharmaceutical Research. So please welcome Professor McCammon to UCI.
Well, it's a pleasure to be here today. I always enjoy coming here and reminding myself what a beautiful campus you have. It's been about a year and a half. I gave a talk to the theoretical chemistry folks, and that talk was more on kinetics of reactions and diffusional processes and things like that. So I thought I'd cover a different area of the territory that we work in today. Perhaps a more practical area of drug discovery. Is this too loud? I'm not sure if the sound level's okay. Okay, good. Let me start by, it's a little odd, but the names are all there. I always like to start by acknowledging the folks in the group who really have done the work that I'm going to be talking about. I often joke, although it's not strictly a joke, that my job as a group leader is all the things they don't teach you about in college or graduate school. I work as a small businessman and an immigration attorney and a psychiatrist and things like that. Meanwhile, these folks are all doing the actual things that I'll be telling you about today. I'm very fortunate to have had and continue to have a very talented group of co-workers. A lot of these folks have moved on to their own faculty or other positions. Nathan Baker was a very gifted graduate student in chemical physics who did enough to get a PhD in about a year and a half, but he had 5 years of support from the Howard Hughes folks. And so he did the equivalent of a second PhD in applied mathematics, and then it was too embarrassing to keep him around, so he joined the faculty at Washington University and recently got early tenure there. So he's done very well. Paul DeBacher is an assistant professor of theoretical genomics at Harvard University. John Monget's up at Stanford now. Jessica, I'll mention some of her work a little bit later. later if time allows. She's on the faculty now at the University of Utah, but moving to University of Chicago next year. Romy Amaro needs no introduction. She's on your own faculty now, and I will be talking a lot about Romy's work, so I'm going to embarrass her a lot today. I hope she's ready. Heather Carlson, I mentioned some of her work too, is professor of chemistry at McCammon at Ann Arbor, University of Michigan. I can go on. The point is that I've been very fortunate to have some very talented people help out with these things. And I'll try to point more specifically to their contributions as I go along. So my own start in this came about during my postdoctoral work with Martin Karplus. At that time, people were beginning to think about motions in proteins. There was still a sense on the part of many people that proteins were essentially static structures. I think that's something that came out of X-ray crystallography. You look at a structure and maybe build a model by using brass rods and little locking bond angle devices. And when we began doing molecular dynamics simulations of proteins, we were, I think, surprised to see how fluid the motion is in these molecules. There's a lot of mobility in proteins. This is a typical set of snapshots that you might get at, say, several picosecond time intervals from a molecular dynamics simulation of a small protein. Molecular dynamics, of course, is just the technique of using an approximate potential energy function to sort of evaluate the forces on each of the atoms in the protein. Coulomb's law, electrostatic interactions, and those forces are turned into displacements simply using Newton's equations of motion, force equals mass times acceleration. So we can determine what the accelerations of these atoms are in any given structure and use that to step the system through time on a computer. The kind of thing that one usually sees is in this superposition here where you have a lot of motion of groups at the surface of the protein and somewhat less in the interior of the typical globular protein. But as I often tell people, and those of you that have heard me talk before will hear some of the same old tired jokes, but it's not entirely a joke. I was starting as a faculty member, as Pierre said, in 1978 at the University of Houston. And you know, my wife was also early in her career. She was doing a neurology residency at the medical school there, and we'd come home after a long day of work and you know, complain about the trials and tribulations of students who were not absorbing the material or patients who weren't taking their drugs or things like that. And then we sit down and do our evening reading, which in my case meant usually pulling out a copy of Physics Today and seeing, you know, what's cooking. And my wife would get out her reading, which was something called Morbidity and Mortality Weekly. That's, as you imagine, a barrel of laughs. But it's a magazine that's still published weekly by the medical community, and it does just exactly what the name suggests. It describes, you know, what people are getting sick from and what they're dying from that particular week. And around 1978, certainly by 79, there were an increasing incidence of alarming arrivals at the San Francisco General Hospital emergency room of young men who, in the prime of life, very vigorous, they would come in with strange patterns of symptoms, oftentimes florid infections of their oral cavities with fungi of various kinds, spots on the skin associated with the skin cancer known as Kaposi sarcoma. These, of course, were the first AIDS patients. And the United States National Institutes of Health responded to the appearance of this emerging disease in a really heroic way. They started a program called the Structural Biology of AIDS program, where they would make targeted grants to universities, medical schools, and even to small or larger companies with a specific goal of trying to find the gene products, the proteins associated with AIDS viruses, and see if one could target those proteins, particularly the enzyme molecules, with inhibitors that would become effective antiviral agents. So a group, again, I was in Houston at the time, but a group of faculty members at UCSD responded to this by starting a small company called Agron Pharmaceuticals. They had a very good crystallographer, Joe Kraut, who's retired faculty member at UCSD. He had some of his postdocs start as a new crystallographic group in Agron Pharmaceuticals. They had very good viral assay people, very good medicinal chemists, but they knew they wanted to use computation as part of this. Computers were beginning to be used in a serious way in drug discovery, but it was still somewhat new and even controversial. But they reached out to me and recruited me to start the computational part of Agron Pharmaceuticals, which I did with a couple of postdocs, and stayed involved with the company for a while. Suffice it to say that turned out to be quite rewarding and quite successful. So again, AIDS was first described as a recognized disease, although its origins were not clear. By 1983, it was clear that it was a viral disease caused by a retrovirus, that is, a virus whose genome is not DNA, but is rather RNA-like material. Molecular biology was used quickly to determine which enzymes were coded for in the viral genome, and there are three of them. There's reverse transcriptase, which is needed to take the viral RNA genome and convert it, reverse transcribe it into a DNA molecule. There's an integrase, which is in some ways the nastiest beast here. The integrase takes the viral DNA, viral-coded DNA, and integrates it into the genome of the infected human cell, typically a T lymphocyte. At that point, that cell is condemned. It's going to have this as part of its genome as long as the cell lives. And that's a big problem even now for thinking of therapeutics for infected patients. The DNA genome, including now this viral piece, is expressed in a normal way with the typical transcription and expression machinery, but now in addition to making human proteins, it makes new polypeptide chains from which new viruses are assembled, and there's a protease, the third enzyme associated with the enzyme, that breaks up this polypeptide into just the right pieces to assemble a new variant, a new viral particle. These are all potentially targets for antiviral discovery, and in fact, reverse transcriptase inhibitors came on stream fairly rapidly because this enzyme, of course, works on nucleic acid. So do a lot of the enzymes that are used to treat cancer. And so AZT, for example, a thymidine analog, was found quickly to be an effective inhibitor of reverse transcriptase and was of some help in treating patients. But still, the death rate was going up very quickly from HIV infections. Agron decided to target the protease because, again, it was known from molecular biology that this is a relatively small protein. It should be fairly easy to isolate and hopefully develop crystal structures so that it could be used for structure-based drug discovery. The integrase has been tougher, and I'll come back to that. So why think about this? Well, even in these early days, it was recognized that proteins are flexible molecules. Molecular dynamics had certainly made that clear, but crystallographers who look carefully at their data know that there's flexibility in proteins. Certainly NMR data was beginning to show that there's a lot of motion in proteins. The aromatic side chains flip around and the tails wag around in a solution. And that, it was clear, might be important in drug discovery, that a small molecule that doesn't seem like it's likely to bind to any one structure, such as the crystallographic structure, might in fact bind to one of these structures that evolves in the course of the fluctuations of the protein structure. Well, that certainly turned out to be the case with HIV protease. HIV protease is actually a homodimer. There are two small peptide chains of 99 amino acid residues that form this flap-like structure that comes down over the active site, where substrate molecules or potential inhibitor molecules bind. And there's a lot of motion of these flaps as you put larger or smaller objects in this active site of the enzyme. So Agron worked on this problem. That story has a happy ending. Agron was one of the first companies to develop what became a medically and commercially important HIV protease inhibitor, this compound Virusept. And when these protease inhibitors came on stream in 1995, the effect on public health was quite dramatic. People were increasingly likely to die of HIV infections until these protease inhibitors came on board. And it's literally true that there are hundreds of thousands of people in the United States who harbor HIV but are going on and leading reasonably normal lifetimes by taking cocktails of reverse transcriptase and HIV protease inhibitors. These are not totally benign. There are side effects with these compounds, but it's made a big difference. Nevertheless, people are still dying of these infections, and that's true out to 2009 as well. And this is usually because the patients have run through the list of possible compounds or compound cocktails and have developed resistance to essentially all the existing compounds. And so there's been an interest in trying to find other targets for HIV and to add to the arsenal of drugs that we can help these patients with. Well, with the work on HIV protease, we were moved in our group to develop what we called a relaxed complex method, which is nothing more than sort of officially describing what I've described in words already. That is, we'll make use of molecular dynamic simulations to sample the conformations of a target protein. We use rapid docking methods, the kind that Pierre has developed here at Irvine, for example, or docking methods from other groups. The Scripps Research Institute has AutoDock, for example, we've used. Here you just take a given structure of a protein and throw a number of small molecules at it in different positions and orientations and see what fits according to a rather crude scoring function. If something looks promising, you can trundle out more quantitative methods to estimate the standard free energy of binding. And if you find something that binds well at one side of a protein, you might look for something that binds nearby and think about linking them together to build higher affinity. So we described this method. I won't spend a lot of time on this because I'm going to run out of time if I'm not careful. We demonstrated this method works on a small protein called FKBP12, which binds the immunosuppressant agent FK506. Here I just want to show that with a relatively short simulation, only 2 nanoseconds of this relatively rigid protein, you do get a lot of motion in the binding site. Here it's rather open, here it's more narrow and pinched off. If you measure the so-called surface accessible surface area, represented by red here, you can see that the exposed surface area fluctuates dramatically, even in a short time, from on the order of 1,000 square angstroms to perhaps 700 square angstroms. And a compound that doesn't fit here because it's a little too large might fit very nicely here. And so this is what we're trying to look for in the relaxed complex scheme. You get the correct position of a compound. Let me move to say something about the HIV integrase problem. HIV integrase is that third enzyme that I said was sort of the really nasty one in the triumvirate of enzymes associated with HIV. And it has 2 distinct functions. It takes a piece of the viral DNA that's been reverse transcribed, cleaves a couple of nucleotides off the ends of what's called the 3' processing step, and then it integrates that into the target DNA, the human DNA, leaving some breaks that are then cleaned up by normal cellular DNA repair enzymes. It's been a horrible enzyme to work with because it's It's a large enzyme to start with. It has 3 domains in it, a big N-terminal domain, a big C-terminal domain, an even larger catalytic domain in the middle. It functions not as a monomer, but probably at least as a tetramer of these things, and probably is not fully active until it's interwoven with some of the nucleic acids that it processes. So it's been a nightmare for structural biologists to make progress in getting structures of the enzyme. The happy thing that happened just 10 years ago was that the David Davies group at NIH was able to get a crystal structure of just the catalytic subunit, just in a monomer form, but with a small molecule inhibitor bound to the surface of the enzyme. And what it looked like was that there's a small local depression in the surface of the enzyme where this Shinobi inhibitor was found to bind. But there were problems with this crystal structure, and David was as a very good crystallographer, was careful to fully describe the problems with the structure, namely in the so-called asymmetric unit of the crystal. There were 2 of these proteins bound face-to-face, so closely together that the inhibitor bound to one protein actually contacted the inhibitor bound to the neighboring protein. That's shown in this figure from the Davies paper. These compounds actually are so close, bound to 2 neighboring proteins, that they form hydrogen bonds across the interface in the asymmetric unit. So this is kind of a red flag saying, be careful, there may well be some effects of crystal packing here. There may be what crystallographers hate to admit, sometimes happen called crystal packing artifacts. So what would happen if we took one of these subunits with the inhibitor bound, pulled it out of the crystal, put it in some salt water, and ran a molecular dynamics simulation on it? What would happen if we tried this relaxed complex scheme? Well, we did that, and we got a big surprise. The big surprise was that this little dimple where this compound binds is still there in the snapshots that we look at, but oftentimes this dimple opens up into a long trench across the surface of the protein. This trench breathes, open and closed, at Brownian motion kind of fluctuations. And Heather Carlson's the one that did these simulations. She's the one who's on the faculty at Michigan now. Julie Shamus just recently finished her PhD work, but Julie's the one that went ahead and redocked the Shinobi compound to some of these snapshots and found that in many of the snapshots, this compound binds approximately where it's seen in the crystal structure, but you find a second binding site in many of these structures, in which the ring that's over here on the southwest is now up here in the northeast. So this molecule has actually rotated and translated by about 20 angstrom units downstream. Well, Julie's a very clever young woman, and she had the idea, well, what happens if you link these together? Might you make a compound that binds more strongly? She proposed using a little naphthalene linker and described what are called butterfly compounds. You have not one wing, but 2 wings. And predicted that these butterfly compounds would bind significantly more strongly, free energy binding, than single wing compounds. Well, we wrote this up and got published, and as I again often say, you work hard, the students work hard, I don't work hard, the students work hard, write the paper, I put the grant numbers on it, and it goes out and gets published. Is anyone ever going to really read this thing? Or is it just going to collect digital dust in the California Digital Library? Well, the good news is that occasionally people do read papers, and in particular people at Merck Pharmaceuticals read the paper. And so very soon after it appeared, I was contacted by a woman named Daria Kazuda, who's the vice president for antiviral discovery at Merck, who came and visited us at UCSD and told us an amazing story, which was that they had been pushing a project to discover HIV integrase inhibitors for a long time and basically had abandoned it because they couldn't interpret their data. They made a lot of analogs of the Shinogi compound. They assumed they bound at this one spot in the protein from the Davy structure. So they measured the strength of binding of these compounds to this integrase catalytic domain. They also did mutations, mutagenesis experiments on the enzyme because they knew that mutagenesis would eventually lead to resistant strains of the virus. They wanted to explore that. But the data that they came back with just completely flummoxed them because sometimes a mutation here would knock out binding in a way that you would expect, but sometimes a mutation here would not knock out binding for some of these compounds. And sometimes mutations elsewhere in the protein would affect binding. They didn't know what to make of it and shelved the project until they saw Julie's paper. And as Daria said, it was like the lights went on. They went back and looked at the results of the possibility that there are 2 binding sites that are active, and all the data fell into pattern. It all made sense. They made the butterfly compounds, and they bind more strongly than the single compounds do. This validates Julie's hypothesis. What Daria and her group at Merck finally decided to do, rather than making butterfly-type compounds, was to look for compounds that could do exactly what you see in this picture, could bind here, and another compound of the same type would simultaneously bind in the second position in the structure. The hope was to try to mitigate the resistance problem. That if you had something, a mutation that knocked out binding at this site, perhaps you would still have binding at this other site and vice versa and retain some activity in the compounds. So Daria and the Merck group wrote this idea up. They were very nice and gave credit to Julie for helping to steer their thinking on this, and moved very rapidly to put a series of compounds into preclinical trials. This compound was written up in their PNAS paper, but was not clinically a candidate for development. This compound, which is very similar, moved into phase 1 clinical trials, but had some toxicity problems. Their second backup compound turned out to be a miracle drug. This MK0518, this inauspicious sounding name, soon became Icentris, or raltegravir, announced at the Los Angeles Congress for AIDS in 2007, was moved very rapidly into fast-track phase 3 trials and very rapidly approved under that program just about 2 years ago, and is now very widely used in AIDS clinics and hospitals around the country. Basically, those few thousand people a year that were still dying of HIV infections, that number has dropped to less than half of that number because of this compound. So computer modeling can make a contribution to this stuff. This is not the stuff of dreams. The Relac complex scheme has been organized by Romy and developed in very substantial ways that I'll clarify in a minute. But again, it starts typically with simulations that give you these snapshots. There are ways that you can improve on picking every 10th snapshot. There are ways you can cluster things to try to get an optimal set of representative compounds that's more manageable from a computational point of view. There are various ligand ensembles you can use for the virtual screening, various things you can do afterwards. I'm not going to really go into that. Here's Romy. First of two more stories. Avian influenza has been in the news a lot recently. Avian influenza and swine flu. They both have a particular kind of protein on the surface that is the one target that people really have for influenza antiviral compounds, the so-called neuraminidase inhibitors. There actually are 2 proteins that bind to these carbohydrate molecules that are on the surface of lung cells and are involved with the way the virus invades these lung cells. Sialic acid here, which is very similar to neurominic acid, is recognized by these cell surface, by these these viral proteins, the neuraminidase and the hemagglutinin, which I'll mention in a minute. They bind to this sialic acid residue, and particularly hemagglutinin binds to sialic acid. The lung cell thinks, something's sticking on my surface, maybe it's good to eat. It pulls this into the cell by an endocytosis process. And the virus uses that basically as a Trojan horse mechanism to get in the cell and then do its dirty thing, leading to many more virus particles in the cell. And then those virus particles have to get out, but they get stuck on the same sialic acid residue, and that's where neuraminidase comes in, which hydrolyzes one of the bonds here and allows the virus to release. So the most effective antiviral drugs for influenza are inhibitors of this neuraminidase. They basically prevent the virus from spreading as rapidly within tissues, and your immune system has more opportunity to combat the virus. So here's this piece of this neuraminidase molecule with a small antiviral drug, this stuff called Tamiflu, bound to it. You've all read about Tamiflu. This is the stuff that's being stockpiled by the metric ton in all developed countries in case of a large outbreak of avian flu or for treating people with the so-called swine flu. Well, it binds right here, and Romy said, let's think about what we can do with relaxed complex ideas. So she did a molecular dynamics simulation of the protein, and cast your eye on these loops down here. It turns out that they move around a lot. Here's some pictures from Romy's first article on this, showing how much motion there is in these loops. Looks like a good candidate for one of these docking and relaxed complex approaches. Tamiflu binds where it should when you use a docking scheme like AutoDock to place this compound back in the binding site, so that's kind of a little benchmark test. When you take the crystal structure or different snapshots from the molecular dynamics simulation and dock a large number of molecules to them, you find that where things like to bind seems to be dependent on the conformation of the protein. Here they're mostly binding around this main binding site, but here the actions kind of move down here because these loops are moving around and creating new pockets and pockets with changed shapes on the surface of the protein. Suddenly here's another area where things seem to like to bind. Maybe there's more here than we realize from the crystallographic structure. So to make a long story short, this virtual screening has led to a set of structures, 27 compounds that we referred to our experimental collaborators, and turns out about a quarter of them have very significant activity in the wet lab. This is work from Ian Wilson's group at the Scripps Research Institute. So these are candidates for further development. So things are going well. We screen, we use this relaxed complex method. There are things you can do to increase the accuracy of the results, but I'm not going to really dwell on that. Let me tell you one other short story and then a couple of more methodological slides about new techniques, and then we'll be done. What's really made Romy famous is this work on African sleeping sickness. African sleeping sickness is caused by a single cellular protozoan, a nasty little bug called a trypanosome, Trypanosoma brucei. There's a number of trypanosomal diseases. This is the one, this bug causes African sleeping sickness. People in a large portion of Africa are outdoors. They get bitten by something called the tsetse fly. A name, once you've heard it, you never forget it, the tsetse fly. Nasty little thing that bites you and injects some of these trypanosomes into your bloodstream. The trypanosomes propagate in your bloodstream and then start invading other tissues, which is really what causes the damage. In the case of T. brucei, these things have a predilection for the central nervous system, and so they go to the brain, they cause stupor, coma, and if untreated, they're invariably fatal diseases. Sleeping sickness because the patients begin to lose consciousness. There's an American trypanosomal disease that's prevalent in Latin America. T. cruzi causes Chagas disease. It's beginning to show up in the United States because of transfusions. It's going to show up a lot more in the United States because of climate change as the insects that transmit that disease become more common over the border. You'll hear more about Chagas disease. Anyhow, I could go on all day about the biochemistry of this critter. It's just like something from another planet. It has whole metabolic pathways that are unknown in mammalian organisms. In particular, the pathways for creating mitochondrial DNA are totally unlike anything in you or me. There's an incredible array of genetic material, DNA maxi circles and small mini circles present in the mitochondria of these organisms. The maxi circle gets expressed into a pre-messenger RNA, and then these mini circles code for what are called guide RNAs that are used in a large, still poorly understood complex called the editosome that works its way across this pre-messenger RNA and replaces more than 50% of it with new bases. Well, there's some enzymes that are needed to carry this stuff out, and among them is this RNA editing ligase, RNA editing ligase 1, whose structure has been determined by Wim Hol and his group at the University of Washington. And there's a group led by Ken Stewart, also in Seattle Biomedical Institute, that's interested in the biochemistry and medicinal chemistry of these compounds. So I ran into them at a meeting in Italy, and this a few years ago, and said, "Hey Ben, what's new? You got any structures that we ought to try and design drugs with?" And he pointed us to this one. And about that time, Romy was getting up to speed in the group, and so very quickly, simulations were done, and compounds were screened. I won't walk you through all this stuff. This just shows if you take a whole bunch of structures equally spaced during a simulation, you can get a certain amount of coverage of the conformational space of the protein. If you select a much smaller number of structures by clustering these guys and identifying things that are most distinct in some sense, you can cover essentially the same ground, but instead of having to look at 400 structures with your virtual screening, here you only have to look at 33 and essentially get the same results. That's Romy's idea. And what came out of this is a set of compounds that not only did we think were good, but when they were tried out on the enzyme in the test tube, the wet bench people said, "Hey, these are good." And so they're moving toward further development. It was described in this recent issue of PNAS. But these are very promising compounds. This work has moved on at the wet bench to compare the affinity of, or the activity of these small molecules. This is one of those 5 compounds I just showed you. It basically kills the trypanosomal enzyme at a submicromolar concentration, which is a good start. It's nowhere near active enough to use as a drug. But when you try it against the closest analog we could find in the human enzyme portfolio, it took more than an order of magnitude more substance to inhibit this human enzyme. So we already have a bit of a therapeutic window where we can hopefully kill bugs, but not the hosts of the bugs. What the plan is now, of course, is to use continuation of this method and other methods to try to make compounds that move to the left in terms of their activity. We'd like to get them down to the nanomolar or less range to broaden this window and require lower doses of the drug to treat the patients with this disease. You can do structures of these things. Water molecules play a role. I'm going to just show you the last few slides to say that not all of what we do is just computer games type stuff. I'm trained originally as a statistical mechanic. I do a lot of funny stuff for a statistical mechanic, but sometimes we do statistical mechanics. And these are formulas for the standard free energy of binding of a small molecule to a protein, or in general, any non-covalent pairing. The reason I mention this is because it's astonishing. You'd think that, this is stuff that would have been done, in the 19th century. People would have figured out in terms of what are called partition functions. These are things you can evaluate on a computer. What the standard free energy of binding should be for a pair of non-covalent molecules. But up until 10 years ago, there was a lot of woolly-headed stuff out in the literature about Craddock entropies and this and that's and mysterious mumbo-jumbo. So Mike Gilson, a very talented postdoc in our group, sat down and did the derivation from scratch and came up with what are now the widely accepted formulas for calculating these standard free energies of binding. This is essentially equivalent to the binding constant. Smaller values of this, if you go from 0 to minus 10 kilojoules per mole, that's a good thing. So these are useful for calculations. Mike, I should mention, has been a professor for some years at the University of Maryland, but he's moving to UCSD next month to join our faculty. Question?
What are the sigmas?
The sigmas are things that have to do with redundancy of orientations. So for a benzene ring, you have a sigma of 6 because you can turn it 6 ways and the molecule looks the same. Statistical factors, they're called. There are simplifications of those formulas that are useful in practice. These were developed by Jessica Swanson, who, as I mentioned, is a faculty member at the University of Utah, now they're joining the University of Chicago, Argonne National Lab next month. And Richard Henchman, who's a chemistry professor at the University of Manchester, now he's a postdoc, she's a grad student of the group. This is the thing I'm most excited about right now. At least one of the two things I'm most excited about. Accelerated molecular dynamics simulation. One of the problems we have with using this relaxed complex method is that molecular dynamics is actually a slow and expensive way of generating conformations, distinct conformations of proteins. And so when Donald Hamelberg came for a postdoc to our group, I said, Don, why don't you do something about that? And Don went away and came back with this idea of accelerated molecular dynamics. The idea is the following. The real potential energy surface for the system is this thin solid line here for this little one-dimensional model that I'm showing here. And if you ran a molecular dynamics simulation starting out in this state here, you tend to just bounce around for a long time in that one state, and only occasionally will a collision with the molecule happen that's strong enough to bounce it over this energy barrier into another state. So why not raise these minima and move around on, say, this dot dash curve instead? That's closer to the top, you'll cross over more easily, sample more conformations in less time. So that was Donald's idea, and you might say, but wait, that's cheating. That's not the real potential energy surface, and you're quite right. It turns out that you can sample configurations more quickly this way. It turns out also that you can happily recover exactly all the equilibrium properties of the system in principle, the thermodynamic and structural properties, because you know how much you've cheated at every configuration, and you simply multiply the weight of that configuration by the Boltzmann factor that contains this energy correction. The thermodynamics comes back exactly, the structural properties come back exactly. What you have lost is the dynamical details of the system. John Mongan's the guy who's at Stanford now, who's a grad student in the group, MD, PhD type. So we published it, and does anyone read these papers? Well, yes. Someone read the paper, and the person who read the paper was Martin Blackledge, a very good NMR person in Grenoble, perhaps the best NMR person on the continent at this point in Europe. And his group was interested in the magnitude of fluctuations in proteins as measured in two different ways. One is the traditional relaxation-based measurements using T1s and T2s and things like that, and so-called Lopari-Szabo analysis, which allows you to get some sense of how much the backbone amide planes, these little planar groups that connect one amino acid to another, how much those planes tilt around on average as a function of time. Well, this measurement gives you a sense of how much motion you have in the protein on the picosecond to nanosecond time scale. And the amplitude of motion is gauged by what's called an order parameter. This is that second Legendre polynomial type thing for the math folks. A value of 1 means that you don't really have any dispersion at all in these angles. They're just all the same from one protein to another, from one time to another. A value of 0.5 means things are moving around pretty substantially from one molecule to another. So on the picosecond to nanosecond time scale, there's a certain amount of motion, but not a whole lot in the core of the protein. This is a newer method, residual dipolar coupling measurements, that in principle allow you to gauge the amount of motion in the protein on the millisecond time scale, much longer time scales, much more biological time scales. And so it's an important advance. These are difficult and even somewhat contentious experiments. If you get add-backs in the same room with Martin Blackledge, you'll have a big argument about exactly how you should interpret these things. But I think it's settling down that if you do things carefully with lots of alignment media and you are careful in treating your Gaussian models for these distributions in 3 dimensions instead of 1 and things like that, you can get a sense that this is probably a good picture. Nevertheless, Martin was worried about this because there's a lot more motion in this structure than there is in this structure. A lot more motion. I mean, here where you had a little bit of a ditzel here, this whole loop now is, just flapping around in the breeze. And Martin said, oh boy, are we going to get beat about the head and neck by the reviewers if we send this out just with NMR data? Isn't there another way that's independent that we can compare with this stuff? Well, Blackledge and particularly Phineas Markwick, a postdoc with him, had read Donald Hamelberg's Accelerated Molecular Dynamics paper, which purports to be able to sample configurations on the millisecond time scale with suitable parameters in that acceleration procedure. And so I should tell you, I have this bad habit of when someone writes a paper in a group and my name winds up on it at all, American Chemical Society, you do a little thing, you put a star by the name of the corresponding author, and that's supposed to be the head of the group or the main cafe or mojo or whatever. And I always let the student get the star because they're the people that did the work. I just put the grant numbers on the paper. So Blackledge assumed that Donald Hamelberg, the little star, was the guy who ran the group and sent him email and said, "Dear Professor Hamelberg, is there some way someday we could get access to your software to try to see what simulations on the millisecond time scale might look like and compare them with these millisecond time scale experimental results?" So Donald said, "Sure, I know Andy's really relaxed about that. Just send him the software over the internet." And I didn't know about all this until I got this paper review. And truthfully, I'm glad I didn't know. It would have scared me to death because what is shown is that the long time scale MD simulation with accelerated molecular dynamics and the long time scale NMR results for these order parameters, if the labels weren't there, I'd have trouble telling which one's which. they pretty much agree in terms of these unexpectedly large amplitudes of motion on the millisecond timescale. For comparison, here's what you see on the nanosecond to picosecond timescale using Lopari-Zawa type methods. And recapitulated in the dotted line in this lower curve is the long time scale stuff from up above on a slightly different system of numbers here. Notice how much more motion there is on the long time scale compared to the picosecond to nanosecond time scale. There's a lot more motion really everywhere through this protein virtually, but particularly in these loops out on the surface. That's why I'm excited about this method for generating snapshots of proteins that would be good for drug discovery. You may find more holes in these proteins that are in rare structures that we can take advantage of with new pharmaceuticals and do something useful with this stuff. But accelerated molecular dynamics has become a big business now, and we're pushing it kind of hard. There's another way you can get the long time scales. I'm just going to race through these. People use this method too, called coarse graining. That is, instead of having simple atomistic models of proteins like is shown here, going to a simplified model in which every amino acid is replaced by a single effective sphere of some kind. You've got to play with the potential function quite a bit. It's never going to be as accurate as atomistic models. But there are some things you can do, because you can do what's called Brownian dynamic simulations, simulate the diffusional motion of these chains in water and move out to very long time scales. But really keeping track of something like the real time of the system, in this case out to a microsecond. And this is work that now, she was a postdoc in the group, and now Professor Chang, she's on the chemistry faculty at UC Riverside, was able to simulate the binding of a drug molecule to HIV protease with this model. And that turns out not to be just an academic enterprise, because the binding rates of these drugs to HIV protease have been measured for the simplest and most familiar HIV protease inhibitors. And Chang was able to show that you get similar rates of binding in the simulations compared to what you have in the laboratory. And so the kinetics of these things may be important. The kinetics are going to be impacted by the environment in which these proteins work. This is sort of the last technical slide, technical as it is, not very. HIV protease really only does its thing when a new virion, a new viral particle, is budding off the surface of a human cell. And within that budding structure, there's almost more proteins and gunk and stuff than there is water there. It's a very crowded environment. And so David Min, a grad student in the group, did a study in which he made these simple models of crowding agents, just spheres with excluded volume, and ran Brownian dynamic simulations of HIV protease in this simplified model in the presence of more or fewer crowders. With no crowders, you get pretty frequent opening events of these flaps on the surface of the enzyme. This is over 20 microseconds of simulation. And with a high concentration of crowders, you can see here kind of crunching in on the enzyme, you get much less frequent opening. So that's, that's something that is right now just academic knowledge, but may come into play down the road in thinking about more effective therapeutics. Last picture, again, I apologize to those of you that have heard me give popular talks before, but I always like to show this. It's just fun. When I teach physical chemistry classes, the students often come up on about the third day or fourth day and say, Dr. McCammon, why all these equations, we're sick of it, equations aren't good for anything, math is hard. And so I like to show them this picture because my dad was a professor of civil engineering, and his PhD advisor, Charlie Ellis, designed the Golden Gate Bridge in San Francisco. And my dad was his favorite student, so when Ellis passed away, he gave my dad all of his books and papers and so forth. And among them was this book on bridge design, which is what he used, chapter 5, to design the cables for the main span of the Golden Gate Bridge. And you'll see there's a section here on wind stresses. If you build a bridge across the Golden Gate where with a good Pacific storm, you can easily get 100-mile-an-hour winds. How much extra steel do you have to put in the cables to offset not just gravity stresses, but wind stresses too? He's gone through, he's corrected some of the equations. Charming preface to this book. This book is from like the late 19th century, and the authors apologized for the length of chapter 5 on suspension bridge design because there were so few suspension bridges. They said they probably will never be very many. And we're sorry that chapter 5 is so long, but we got carried away by the beauty of the mathematics. Well, Ellis found that it's useful. And these are basic physics equations, just like Newton's laws. And using a little math, using a slide rule in his case, you can design something that might be helpful to humanity and build it, and it'll work. And we're just trying to do the same thing at the molecular level. So again, these are the folks that did the work. I'll stop there and thank you for your attention and try to answer any questions if there's time for that. Doug?
First of all, thanks a lot for the beautiful talk. My group and I, we often discuss coarse grain models, and one of the things that I always tell them is that you should be very careful when you see people using them to interpret dynamics and kinetics. And the reason is the mass distribution is different. You have to be very careful treating the friction, and et cetera, et cetera. So I'm wondering what's the secret then in how you were able to get accurate kinetics for the simulation of the binding of the inhibitor to each proteins?
Yeah, I think it's probably because, number one, we're working in an over-damped regime, clearly, so we're really in a Brownian regime, so mass things pretty much go out the window. But beyond that, I think what we're looking at is sort of the simplest kind of polymer dynamics process where sort of the sizes of the beads and the fairly substantial flexibility of the chains is probably governing a lot of what we see in the dynamics of the system. And there's just enough reality in the virtual bond parameters that we use to confer a little bit of resistance to change in a certain preferred structure. So this is kind of like almost a perturbation approximation to reality, I think. I can easily imagine doing calculations with this model where you're not going to get something sensible out of it. So you're right, you've got to think about the applications and details of the applications, and a method like the one I've described, may be useful for some problems and just dangerous for others. It's good to be cautious with these things.
On your accelerated molecular dynamics, where the bottoms of the wells basically got raised, it looked like, at least in that two-dimensional diagram, that they were getting raised asymptotically to the same level.
Yeah.
And I'm wondering why they're not raised asymptotically or proportionally to the depth of the well.
Yeah. You could do that. You could do that. The method that we used, and this is not the only method, we started with this because one of the predecessor methods in the literature that had been described for condensed matter simulations in the physics and materials community had described a method that used these perfectly flat approximations, these truncations of the potential at a particular selected value of the potential energy. That was not something that was pleasant to contemplate during molecular dynamics because the forces become highly ill-behaved where the gradients are not constant and the potential. So Donald's first idea was, well, if we want to do molecular dynamics and not have all the apparatus that went with doing these flat things, was to do these tapered potentials, just curving off from that. Donald has this colorful expression. The original method from the group at Argonne that developed it was puddle skimming. As you can see, you're jumping from one rain-filled puddle to another rain-filled puddle. And so Donald likes to call this the snowdrift method, where like soft snow coming up around the edges of the potential gives you a nice continuous curve. But in fact, you can change the potential any way you want to, as long as you keep track of what you've done for every configuration. And we have various flavors of this now where instead of raising minima up, we bring the maximum down, and that has some advantages for some problems. And there's various other angles you can work this thing. We're starting to do this on lipid bilayers, and the code is free. Anyone that wants it, I think it may help with the. We've done some work with 2 different kinds of lipids and shown that if you start with separated lipids, that they mix a lot more quickly with a little bit of this stuff in it. So it might help with sampling there too. One more question?
Yeah, just related to this, could you just sort of shed some light just to unite us? How would you just add it to the bottom of the well, but not on the heel?
Yeah, well, there are different ways to do it, and one of the advantages of this is that you can apply the adjustments to any term in the Hamiltonian you want to, or more specifically in the potential energy function. So in one case, the first case, we just changed the dihedral angle functions, for example, but not non-bonded interactions. In other cases, you might change the non-bonded interactions in one way and the dihedral angles in another way. People might ask, why don't you just raise the temperature? The problem is that that's a very non-selective way of knocking your system around. And here you can really do fine adjustments to any part of the potential function you want to. Thanks again.
No further questions?